today's class actually is linkage and crossing over. It's a genetics class which has been allotted to me. So essentially, you people must be aware of Mendel's laws of genetics. Mendel was considered the father of genetics, and he gave certain laws, three important laws. The first one was the principle of dominance, that you have a tall pea plant when crossed with a dwarf pea plant. The first generation which you see is all tall. Then in the second generation, what you see is that in the ratio of 3:1, you have a pea plant appearing with the dwarf phenotype in the one ratio. So this means that the character which is controlling the tall plant, that the plant is tall, that is dominant over the character which is a dwarf. And the resulting, this was the principle of dominance. Then, then he saw that in the second generation, the first generation they were all tall plants, and in the second generation suddenly he saw that the dwarf plant appeared, and because the dwarf plant appeared, he said that there are two factors, one for tall and one for dwarf, one for tall and one for dwarf. Both of them, they segregated. So they segregated, and in the second generation you see that because of that segregation you have the dwarf plant coming up. So this was the principle of segregation. And when he used two characters, one was yellow and one was green. I'll explain this with my slides. So you people have read this for a long time. I am presenting my entire screen. সাদাটা দেখতে পাচ্ছ?
Yes, sir.
Totally white? Okay. So if people have got problems, please ask. Okay. So I'm starting. The name of the topic is linkage and crossing over. So you see, first of all, you see a green chromosome. Are you able to see this green chromosome? Yes or no?
Yes,
Yes,
sir.
sir.
And you are able to see the red chromosome? Red দেখতে পাচ্ছ না?
Yes,
Yes,
No?
sir.
Okay, okay. So please talk in between. Please interact with me. That is very important. So it is assumed that in all of your cell, you have got one from your father, one from your mother. Say the green is from your father and the red is from your mother. This is this is the maternal component, and this is your paternal component. One from your father, one from your mother. And during cell division, one single strand, one single green line, this green line, it is known as chromatid, and two of them together they duplicate before the division, and they are chromosome. So each of these green chromosome or each of this red, they are this green and green, they are known as sister chromatids, because they duplicated from only the one thing. And the red one, which you got from your father, the red and the green, although in the same place you have got the same type of genes, but still one is from your father and one is from your mother, they are known as non-sister chromatids. So during meiosis, there is a phenomenon, meiosis one, a phenomenon takes place, and that is known as crossing over. And there is exchange of DNA between the non-sister chromatid. As you can see that over here, a crossing over is taking place, and this resulting structure is a chiasma. This chiasma, as the result of that, you have little portions. say from your father coming in this chromosome of your mother, and little portion of mother coming in this chromosome. They have exchanged DNA. Now they will break up into single chromosome. You can see four strands. They'll be components of four cells. They will be there will be four sperms. So you have one sperm which has got all sperm or egg, whatever you say, it's got one completely green, one has got a mixed with red and green. Another will have mixed with red and green, and one will be completely red. So the completely green and the completely red, they are known as the parentals, and the uh, one which is mixed, they are the recombinants. So you have both the parentals coming as well as their recombinants coming. So now let us go to this principle of independent assortment. Mendel said, see, he took a pea plant which was round yellow. It was a homozygous. Homozygous means it is got round. All the genes are for round. It is not a heterozygous. So actually, homozygous means if it's for round, then both the genes are for round. And if it is yellow, then both the genes are from yellow. Both the genes means one from your father, one from your mother. As you have seen that one comes from your father and one comes from your mother. Both of them are for round or for yellow. So they are homozygotes. In a heterozygote, maybe one is for round and maybe one is for wrinkled. Then it is a heterozygote. Is it clear? Yes, no?
Yes, sir.
Yes,
Okay.
sir.
Yes, sir.
Okay. And then you have wrinkled green, which is also homozygous. So if you have problems, please immediately interrupt me in any language you want. Okay. So in the F1 generation, you see that all of them are round and green, round and yellow. They are round and yellow. You see, this is round and yellow. This is wrinkled and green. So in the F1 generation, both of them are there. That means round is dominant over wrinkled and yellow is dominant over green. Now they are crossed with themselves. And in the what you find that a new ratio of 9 is to 3 is to 3 is to 1 ratio coming. And you find that two new traits, that is yellow and wrinkled, which was not there, it came up, and a round green, which this also was not there, that also came up. Apart from the parental ones, that is which is the yellow round and the green wrinkled, you had this new thing came, and this means that this used to come in the ratio of 9 is to 3 is to 3 is to 1. And this is because this new character comes, and because of this he gave this law known as independent segregation of two traits. The two traits are, one was round and the other was yellow. Both the traits, one was the shape of the pea pod and the other was the color of the pea pod. So because of the two traits, whether it's round or whether it is wrinkled or whether it's yellow or whether it is green, it was independent, because independently these two new combinations came, and it came in the ratio of 9 is to 3 is to 3 is to 1. So Mendel's law of independent assortment, it states that alleles of two or more different genes. So what are alleles? Alleles are contrasting form of the same gene. What are the contrasting forms of the same gene? Say for example, the gene is for shape of the pea pod, matar ka dana ka shape. The shape of the pea pod, it is either round or wrinkled. So round is an allele, wrinkled is another allele. It is contrasting form. एक गोल है, एक थोड़ा सा कोकड़ानो है. So round or wrinkled. So these are the two traits. So these are the alleles. So the alleles of two or more different genes get sorted into gametes independently of one another. In other words, the allele a gamete receives from one gene does not influence the allele received for another gene. So the gene is for shape of the pea pod. The alleles is round or wrinkled. So each of this will have a gamete, a pollen, and this will have another. You'll have the ovule in the pea plant. So each of the gamete will get either a round or a yellow. Here it will get a wrinkled and a green. So this will be both round yellow and wrinkled green. Their gametes will have either round yellow, wrinkled green. So the presence of either round does not, if it is round, it does not influence. Listen very carefully. It does not influence whether the shape will be wrinkled or not. So that means it is independent. ये नहीं कि हमेशा अगर राउंड रहेगा तो उसके साथ येलो रहेगा, या रिंकल्ड रहेगा तो उसके साथ ग्रीन रहेगा. हाँ, तो रिंकल्ड होने से ये कभी किसी भी तरह राउंड होने से रोकेगा नहीं. दिस वाज व्हाट मेंडल सेड. ठीक है. एंड यू हैड अ रेशियो ऑफ 9:3:3:1. इज इट क्लियर? না
Yes,
হলে
sir.
Yes, sir.
ওকে ওকে, না হলে না বুঝলে বলো. But this particular experiment was done in sweet pea, Lathyrus odoratus, and that was linkage. Mendel was very fortunate that round, the character round or yellow, that is for the shape of the pea pod, and the whether the pea pod is round or wrinkled, or whether it is yellow or green. The yellow and green, they were characters, the shape and the color, they were on different chromosomes. Now we are talking about chromosomes. He was unaware about that. He called them as factors, but they were on different chromosomes. So this independent assortment, the presence of yellow did not hamper whether the pod can be wrinkled also. কোঁকড়ানো হবে কিনা yellow উপরে কোনো নির্ভর করেনি. But in an experiment in sweet peas, something else was seen, and that was linkage. So linkage was first. What is linkage? Link means one is joined with each other. It could very well be that the presence of yellow did not made always yellow was there always with round. It did not allow wrinkle to come. Because yellow and round, they are linked together. পাশাপাশি হলে কি হবে? দুটো জিন যদি পাশাপাশি হয়, if they are beside each other, then what is the problem? The problem is that they will not get separated. So linkage between genes was separated during crossover. उसीलिए first I showed the picture of crossover. So linkage between gene was first discovered in experiment with sweet peas. This is the flower of a sweet pea. So there was some deviation from independent assortment. That 9:3:3:1 was not seen. This was found by Bateson and Punnett in 1905. That famous Bateson and Punnett square, which you people, statisticians use. He was basically a statistician. So he took one. The pea plant had traits. Sweet pea. Flower color can be either purple or red. This is either purple or red. You had seen a purple flower before, or it could be red. Purple is dominant to red. अगर purple है तो it will suppress this red color. And the shape of the pollen, it can be oblong. Oblong मतलब लंबा, or it can be round. Oblong is dominant to round. So यहाँ पे दो चीज ले रहे हैं, shape of pollen and flower color. Flower color दो color है, two types of flower color. One is purple, one is red. Purple is dominant to red, like tall is dominant to dwarf, purple is dominant to red. And pollen shape लंबा भी हो सकता है और गोल भी हो सकता है. हाँ, pollen male reproductive gamete होता है plants का. So pollen can be either oblong or round. Oblong is dominant to round. So considering this, so what was the deviation? So what he did was Bateson and Punnett in 1905. He took purple long, capital P, capital A, capital A, पर्पल लॉन्ग, लंबा और पर्पल फ्लावर नॉर्मल जो दिखता है. सो दो गैमेट रहता है, वन फ्रॉम फादर वन फ्रॉम मदर, वन पी फ्रॉम फादर वन पी फ्रॉम मदर, दो क्रोमोसोम सोचो, एंड वन एल फ्रॉम फादर वन एल फ्रॉम मदर, पर्पल लॉन्ग. इट वाज क्रॉस्ड विथ रेड एंड राउंड. सो पी होता है, कैपिटल पी होता है पर्पल एंड स्मॉल पी इज फॉर रेड, एल फॉर कैपिटल एल फॉर लॉन्ग एंड स्मॉल एल फॉर राउंड. So they were crossed. F1 ऑफस्प्रिंग जैसे दिख रहे हैं, P इज एक कैपिटल P इज डोमिनेंट ओवर स्मॉल p, सो वो पर्पल हो गया, रेड के ऊपर डोमिनेंट है, सो पर्पल भी हुआ, ये लॉन्ग भी हुआ, बिकॉज लॉन्ग इज डोमिनेंट ओवर राउंड स्मॉल एल के ऊपर. नाउ दे वर सेल्फ, पर्पल फ्लावर ये दोनों को क्रॉस किया. पर्पल फ्लावर्स वर क्रॉस्ड विथ रेड फ्लावर्स, पर्पल लॉन्ग विथ रेड राउंड. ये छोटा छोटा पोलन दिख रहा है. आर यू एबल टू सी दिस पोलन ओवर हियर, स्मॉल पोलेंस?
Yes, sir.
Yes,
और
sir.
यहाँ पे लंबा पोलन, हैव यू सीन द लंबा पोलन हियर?
Yes, sir.
Okay, very good. So people are not blind. Very good. This is very nice. Okay, now they were crossed, selfing हुआ, and the first F1 offspring, they were all capital heterozygous. Capital P is dominant over small p, and they were all purple and long. Yeah, for F1 generation में there is no controversy. Now they were self. इंडिपेंडेंट असॉर्टमेंट होता तो क्या होता, 9:3:3:1 रेशियो में होता. बट F2 क्रॉसिंग किया, पीपी कैपिटल P स्मॉल p कैपिटल L स्मॉल a, ये दिस क्रॉस वाज डन. तो गैमेट्स क्या हुआ, कैपिटल P कैपिटल L एक गैमेट हुआ, और एक गैमेट क्या हुआ, कैपिटल P स्मॉल l कैपिटल P स्मॉल l गैमेट हुआ. गैमेट मतलब फ्रॉम योर सेल आइदर स्पर्म और एग्स, दे आर हैप्लॉइड. डिप्लॉइड से हैप्लॉइड बन रहे हैं. ओके ये डिप्लॉइड दिख रहे हैं, तो यू आर सीइंग डिप्लॉइड. नाउ दिस इज बिकमिंग हैप्लॉइड. सो ये चारों में, इसमें से दोनों में से एक जाएगा, ये दोनों में से एक जाएगा, फिर ये दोनों में से एक जाएगा, इस दोनों में से एक जाएगा. सो व्हाट आर द कॉम्बिनेशंस फॉर्म? वन कैपिटल P, वन कैपिटल L, वन गैमेट, वन कैपिटल P, वन स्मॉल l गैमेट, वन स्मॉल p, वन कैपिटल L गैमेट, and one small p and one small l gamete. Clear? Yes, no?
Yes, sir.
Yes, sir.
Very good. So 4 different types of gametes are formed. F2 crossing. अब ये दोनों में assortment होगा. So purple flowers, they were crossed, self fertilization, खुद ब खुद fertilize हो गया बस. So these are the colors which you are supposed to see. These are the phenotypes. Purple flowers, long pollen, that is the one parental. पर्पल फ्लावर्स राउंड पोलन, ये न्यू कैरेक्टर. पर्पल के साथ आ रहा है कौन सा, राउंड. पहले लॉन्ग था, अब राउंड आ गया. देन यू हैव द रेड फ्लावर, लॉन्ग पोलन. रेड फ्लावर देखो, रेड फ्लावर मीन्स स्मॉल पी एंड अ लॉन्ग पोलन. दिस इज ऑल्सो अ न्यू कॉम्बिनेशन, एंड दिस वॉज द रेड फ्लावर राउंड पोलन. दिस द पुराना मतलब पेरेंटल जनरेशन. सो एक्सपेक्टेड नंबर क्या है, 9:3:3:1. So 240:80:80:27. This was an expected in the cross. If you add them totally, this was what was expected. That was the ratio of 9:3:3:1. This was what was expected. अगर 9:3 total flowers में 9, this was the expected. But observed was 296 मिला इसका instead of 240, 19 मिला instead of 80, 27 out of instead of 80, and 85 instead of 27. So these were the new recombinant types. ये थोड़ा सा कम आया, and the parental types was 240 and 27. ये वाला parental type. This parental type, इसमें बहुत ज्यादा parental type आ गए, more parental types. So the parental types were overrepresented, and the ratio was 15:6:1:1.4:4.5. भाई ऐसे तो होना नहीं था. as per independent assortment. It was supposed to be 9:3:2. So this was a deviation mila. So the observed F2 generation ratio is 15:6:1:1.4:4.5. Expected ratio 9:3:3:1 is not seen. Parental phenotypes were over represented. पेरेंटल फेनोटाइप्स क्या था, जो पेरेंट्स में जो फेनोटाइप था, दैट वाज पर्पल लॉन्ग, एक पेरेंट था. द सेकंड पेरेंट वाज रेड एंड राउंड, रेड कलर एंड राउंड. 9 it was found in over represent, बहुत ज्यादा মিলা, আরো বেশি পাওয়া গেছে, যতটা আরো কম থাকার কথা ছিল. আর মাঝখানে যে নতুন যারাদের আসার কথা ছিল, the new one that was purple round and red long, this was also ये भी कम था. नॉन पेरेंटल फेनोटाइप्स वर अंडर रिप्रेजेंटेड. तो इसका मतलब इंडिपेंडेंटली ये लोग असॉर्ट नहीं किया. असॉर्टमेंट मींस मिक्सचर, मिक्सचर बना रहे. मतलब झाल के साथ मतलब तीखापन के साथ मीठापन के साथ खट्टापन के साथ तीतापन, ये देर वाज, जो इंडिपेंडेंट होना था नहीं था. तीता के साथ हमेशा खट्टा आ गया. Something like that, if you imagine. So this was the evidence that the two genes do not assort independently. Mixture independently nahi bana hai. So the nature of their physical linkage, physically kya hua? Hum log to statistics, we are looking at statistics. The statistics where it's not very clear. The statistics is very not what is happening inside the cell. So the nature, ye log, at that time we did not know about chromosomes, that the genes were located on chromosomes. We were not aware at that time. So physically these genes are linked, ye pata nahi tha. So the genes are physically linked on the chromosome. So this purple color and long pollen, the shape of the pollen and the color of the flower, both of them actually they were located on the same chromosome. Same chromosome matlab on the same chromosome, pass mein pashapashi duto gene located. They were located side by side. Result is when this crossing over took place, chance that दोनों एक ही साथ दूसरे गैमेट में आ गया. They were always inherited together. एक को खींच के रखता था, एक कैरेक्टर और एक कैरेक्टर को खींच के रखता था, इंडिपेंडेंटली असॉर्ट करने नहीं दे रहा था. If it was on separate chromosome, they were not physically attached. So independent assortment, Mendel was fortunate, he got independent assortment from there. But here he was not fortunate and he did not get independent assortment. So the genes are physically linked on the same chromosome. And linkage is the close association of genes. What are the genes? The gene for the color of the flower, purple or red. Purple एक allele है, red एक allele है. Okay. And long is and the shape of the pollen is of one gene, another gene. Long एक allele है and round is one allele of that gene. Be very clear about what is allele. Okay. Linkage is the close association of genes or other DNA sequence. What is gene? It is a sequence of DNA. Chromosome में क्या रहता है? DNA. So what sequence of DNA is one gene. So ये दोनों DNA क्या, like एक धागा में genes खड़ा बैठा हुआ है, like in on the chromosomes. The closer the two genes are to each other on the chromosome, the greater the chance that they will be inherited together. পাশাপাশি থাকলে ওদের চান্সটা কি, তারা একসঙ্গেই যাবে. কেন একজন আরেকজনকে টেনে থাকবে. একটা সুতোর মধ্যে কি, তুমি সুতোকে কাটছো, হ্যাঁ, সুতোর মধ্যে ধরো দুটো দাগ থাকলে, টানলে একটা নীল দাগ একটা লাল দাগ, যেই টানলে, হ্যাঁ, চান্সটা কি, যেই সুতোটা কাটলো নীল দাগ আর লাল দাগটা একসঙ্গে জোড়া থাকবে. নীল আর লালের মাঝখানে কাটার চান্সটা কম. হয়তো মাঝে সাঝে তুমি 10 বার কাটবে, তুমি দেখবে যে 8 বার তুমি দেখবে যে লাল আর নীল একসঙ্গে, হয়তো 2 বার তুমি দেখবে যে লাল আর নীলের মাঝখানে কেটে গেল. সুতো টানছে, দুটো দাগ দিয়েছে সুতোর মধ্যে, যদি পাশাপাশি থাকে। তো ক্লিয়ার, এটা বুঝতে পেরেছো?
Yes, sir.
ওকে
Yes, sir.
Yes, sir.
ওকে ওকে। ছেলেদের গলা পাচ্ছি না।
জি স্যার।
হ্যাঁ হ্যাঁ, মানে বুঝতে পেরেছো?
হ্যাঁ স্যার।
আচ্ছা, তো এইটাই হচ্ছে ডিভিয়েশন, এইটা হচ্ছে ফার্স্ট লিঙ্কেজ, এভিডেন্স অফ লিঙ্কেজ, linkage, recombination and crossing over. So now let us look in the terms of chromosome. এটা parental, red long, red gene, long gene, আর দুটোতেই ক্যাপিটাল আর ক্যাপিটাল এল, ক্যাপিটাল আর ক্যাপিটাল এল। It is crossed with একটা parental white and short. It is something else. This long is the height of the plant. এটা we are talking about another character. White and short. We are talking about linkage, recombination and crossing over. Small r. So this is a parental which is homozygote. This is a recessive parental, white আর short. লাল ফুল সে জায়গায় তোমার সাদা ফুল, লম্বা গাছ বেটে গাছ, ঠিক আছে, বা লাল চোখ, যাই হোক সে আর যেতে হবে না, এইভাবেই যাই। এবারে F1 জেনারেশন, হোয়াট ইউ ফাইন্ড, ওয়ান প্যারেন্টাল, এইটা এসে গেল ক্যাপিটাল আর ক্যাপিটাল এল, আর এখান থেকে একটা এলো স্মল আর স্মল এল। দিস ইজ দ্য প্যারেন্টাল জেনারেশন। ক্রসটা হলো, এটার থেকে একটা গেল গ্যামেটে, এটার থেকে একটা গ্যামেট এলো, এদের সেলফিং মিওসিস টেক্স প্লেস। ইফ এদের মধ্যে क्रॉसिंग ओवर হচ্ছে কি হচ্ছে না, क्रॉसिंग ओवर वी हैड सीन वेदर क्रॉसिंग ओवर या इन दोनों के बीच में हो रहा है या नहीं, इफ क्रॉसिंग ओवर इज टेकिंग प्लेस बिटवीन देम और नॉट, नो क्रॉसिंग ओवर, दिस एज सच कम्स ओवर हियर। देखो सेम आ गया, क्रॉसिंग ओवर आ गया, यहाँ पे क्रॉसिंग ओवर आ गया, क्रॉसिंग ओवर हो रहा है। रिजल्ट क्या हुआ, F1 का जो गैमेट्स, द गैमेट्स विच आर फॉर्म्ड इन F1, one is this A gamete, capital R capital L, and one is small r small l. This A, यहाँ पे कोई recombination नहीं हुआ, no crossing over take place. So this is known as non-recombinant chromosomes. And now here you have a crossing over taking place. क्या हुआ, कैपिटल R स्मॉल l, ये कैपिटल R आया, ये स्मॉल l आया, क्लियर, एंड यहाँ पे स्मॉल r आया, कैपिटल l. These are known as recombinant chromosomes. Clear? Yes, no?
यस सर।
Yes, sir.
Yes,
Very
yes,
good.
sir.
Okay. So genes that are on the same chromosome travel through meiosis together. However, alleles of chromosomally linked genes can be recombined by crossing over. Nor iska matlab, what does this mean? That the genes here, the capital R and capital L, that are the R and L, the color and the length, height, they are on the same chromosome, एक ही साथ मियोसिस में ट्रेवल किया। So R and L and small l, they travel together in meiosis, एक ही साथ ट्रेवल किया, they have traveled together. But when crossing over takes place, this recombinant combination can take place through crossing over. This is the meaning of this line. Now, next term is we have to talk about what is linkage phases, coupling and repulsion phase. Capital R capital L के साथ small l, this is known as coupling heterozygote. जो इस तरह से है, this is a coupling heterozygote. And capital R small l and small r capital L, this type of a heterozygote, this is known as a repulsion heterozygote. Clear? Yes, no?
Yes, sir.
Yes, sir.
पेरेंटल वाला ये कपलिंग हेट्रोजाइगट है, रिपल्शन वाला ये रिपल्शन हेट्रोजाइगट है। लिंकेज एंड फ्रीक्वेंसी ऑफ रिकॉम्बिनेशन। Now capital R capital L, small r small l, small r small l, small r small l, white short के साथ this heterozygous, this is a coupling heterozygous. इस with a recessive heterozygous homozygous के साथ क्रॉस किया व्हाइट शॉर्ट के साथ. ठीक है. Now what happens? Red long, one is this, one is this. All the four phenotypes, जो 4 combinations बन रहे गैमेट के साथ, this is the F2. रेड लॉन्ग हुआ, व्हाइट शॉर्ट हुआ, रेड शॉर्ट हुआ, व्हाइट लॉन्ग हुआ. ये नीचे वाला कांस्टेंट रहेगा, nothing is happening. But ऊपर वाला, because you had seen 4 types of gametes was formed. This was the recombinant gametes, this was the non-recombinant gametes. अब इसमें तो सिर्फ एक ही टाइप है, একরকমই রয়েছে, only one type is present. So this is same. And since R is dominant over small r, so the phenotype is red. L is dominant over long, so long. यहाँ पे कोई dominant नहीं है, so it's white and short. यहाँ पे you have the new combinations which are coming place. So this is clear. যেটা ঘুরলো?
Yes, sir.
Yes, sir.
Okay. So what was found that out of total of 1000 plants which came in the F2 generation, 450 and 470 were either, this was the parental, ये भी एक parental है, ये भी एक parental है. They were white short and red long. 450 parental, 470, so there were 920 parental. And the recombinant बीच में जो क्रॉसिंग ओवर हुआ है, ये नीचे वाला कॉमन है, लेकिन ऊपर में देखो, this was a different red short and white new combinations. 80 recombinant है. Now you are trying to find out what is the distance between those two genes. One thing was sure, a number of recombinants is less than the number of parental. Do you agree with me or not? Yes, no, agree, no, not agree. Agree করেছো না agree করোনি? বলো.
একবার বলুন কি বললেন?
এই যে নাম্বার অফ রিকম্বিনেন্ট, এটা তো প্যারেন্টালদের থেকে কি বেশি না কম?
Sir, কম?
ডেফিনেটলি
কম, sir.
কম. তার মানে এদের 1000টা গাছের মধ্যে মাত্র 80টা গাছ রিকম্বিনেন্ট হয়েছে, and out of 1000 plants only 920 plants are only the 920 plants are parental. Now you are trying to find out the distance between R and L, red and long, on the same chromosome. You are mapping a gene. So frequency of recombination is 80 divided by 80 plus 920, that is 80 divided by 1000, that is 0.8. And actually the distance between R and L is 0.08. This is the map distance. The frequency of the recombinants which you get is actually the map distance between the two genes. Clear?
Yes, sir.
Yes, sir.
So
this
what
is a
will be the unit?
The unit is normally people use centimorgans, but you don't need an unit. They use centimorgans.
Okay, sir.
Named after T.H. Morgan, Thomas Hunt Morgan. So Morgan, Sturtevant, actually it was Alfred Sturtevant who made this mapping system in Drosophila. The now you are studying the 2-point cross. Next we will, in your exam you will get a 3-point class. In the next day's class we will discuss about 3-point crosses. So the key point of today's class is linkage between genes is detected as a deviation from expectation based on Mendel's principle of independent assortment. ঠিক আছে, এটা মানে ডিভিয়েশন যেটা প্রথমে পড়ালাম. The frequency of recombination measures the intensity of linkage. So যত recombination, the number of recombinants, the lower the number of recombinants, the more close the genes are together. আরো পাশাপাশি যত বেশি বহু जितना ज्यादा recombinants, the genes are more further away from each other. And if you get 50% recombinants, then it is independent assortment. It is located on different chromosomes. So the frequency of recombination measures the intensity of linkage. কত কাছে কি কত দূরে. In the absence of linkage, this frequency is 50%. তার মানে independent assortment এতে you will get 50%. 50% recombinants, 50% parental. If there is not, if there is independent assortment, but for very tight linkage, মানে একেবারেই পাশাপাশি zero, মানে recombinant এলোই না. Clear?
Yes, sir.
Okay.
Yes, sir.
So this is end of today's class. Now I would like to take, if you people have got some questions, please feel free to ask. প্রশ্ন জিজ্ঞেস করতে পারো, জিজ্ঞেস করো। আছে কোয়েশ্চেন কারুর? ইয়েস নো?
নো স্যার।
ওকে,
No, sir.
No, sir.
ঠিক আছে। পরের ক্লাসটাও একটা অনলাইন নেব, কিন্তু অন্য একটা টাইমে নেব, কিন্তু পরের স্যাটারডে আমি অফলাইনই ক্লাস নেব।
আচ্ছা, আজকে তো ক্লাস নেবেন না অফলাইন?
না, আজকে অফলাইন না, আজকে এটা দেখানোর জন্য প্রজেক্টরটা রেডি করে রাখতে হতো, গতকাল ভুলে গেছি আমি।
ঠিক আছে স্যার।
প্রজেক্টরটা রেডি করে এখন সকালবেলা 10টার সময় কাউকে পাবো না প্রজেক্টর রেডি করার জন্য। কোথায় আছে আলমারি থেকে বার করতে হবে, এসব করতে হবে, কাল সন্ধ্যেবেলা বেরোনোর আগে ভুলে গেছি। তাই জন্যই অনলাইন, কারণ এটা না দেখালে তোমরা বুঝতেই পারবে না। সে হাতে হাতে তোমাদের বোঝাতে, সে মানে এইটা বোঝাতেই আমার সে মানে 2 ঘন্টা লেগে যাবে। ঠিক আছে, ওকে,
Thank you, sir.
ইউ
Thank you,
ওয়েলকাম।
sir.
Thank you, sir. Thank you, sir.
Thank
Thank you,
Thank
sir.
you, sir.
ওকে, ওয়েলকাম।